Hello everyone, and welcome to the subsection 2.3 of section 2. So in this video we are gonna decode the genetic code to read the DNA sequence and to identify the amino acids which are present in the polypeptide chain. Before we start, let's just look into what you mean by codons. Codons are basically three-letter word, and there are about 61 different codons which code for different amino acids. Please remember there's just one start codon, which is AUG, which codes for methionine, and there are three stop codons. When we will look into that chart, I'll explain to you what are the three stop codons. For now, let's look at this particular question. They have given you an mRNA, so please remember the codons, the genetic codons will always work with mRNA codes. Will never work with DNA codes. Why? Because DNA doesn't have U, and when you check the DNA or the codon thing, the genetic code, you will see that it always has a U, it never has a T. Anyways, so if this is your mRNA codon, please, the first codon that you will look for is the start codon, which is always methionine, which is always AUG. So once you go on looking for your AUG, the moment you see your AUG, you start your protein translation there. So this is how we start. So we have AUG, the first one. So this is, sometimes they will give you the genetic code in the form of a circle. Sometimes they will give you like this in the form of a table. So when you come in there, let's find AUG from here. So AUG is, A is the first letter, U is the second letter, and G is the third letter. So now you know what is the amino acid in methionine. Similarly, if you have a table here, so this is the first letter A and then U. Then AU, so this is the row and this is a column. You will come to this box, and AUG is methionine again. So we know AUG codes for methionine. The next codon that we have immediately after G, we have AUC. Always look for three letters. So AUC is the next one. If you look here, AUC is isoleucine. So we know isoleucine is the next amino acids. When you come to here, again, for AUC, A is the first letter, use the second letter, so row and column this one. You come to AUC, you know it's isoleucine. Okay, similarly when you go to the third one, which is UCG, just immediately after these three letters, when it ends, you have to immediately catch the next three letters, which is UCG. When you come here, U first letter, C second letter, G third letter, you know it's serine. Okay, so we go here, we know it's serine. Again, we will check with UCG here. U is the first letter, C is the, so you come to this block, UC, and then you start looking for G, and we know it's serine. Okay, so and as I was talking about stop codon, let's look UAA. U first letter, A second letter, and A third letter. See, there's stop written here, so it's a stop codon. Doesn't code for any amino acid. So as I said, there are three stop codons: UAA, UAG, and look, we have another one here, which is UGA. These three will not code for any amino acids. They are stop codon. The moment they are there, your protein translation stops. Okay. And for this genetic code, please remember, genetic code is something which is universal. It's not there for one species or one animal. It's there for everything on this earth, okay? More or less everything. Now, let's practice some MSAT biology questions, which is based on this. The first one is as I said, in the form of a circle. And this time they say that it's a simple one. This time they say they have not given you the codon. This time they have given you the amino acids. You have to start figuring out from there. The first one is valine. So when you go there, let's look. We have GUG. So G first letter, U, G. Okay, that is valine. So GUG is valine. This is also valine. This is also valine. And then GUC, GUC is also valine. So both these options are correct. The next one is AGC. So when you come to AGC, and then you have AGC. So AGC is serine. Okay, that is also correct. So AGC is serine. And then here another one is AGU. So AGU is also serine. So AGC, AGU are both serine. Okay, that doesn't solve our problem. Then you have proline. When you go to proline, we see it's C. So the next letter we have is CCA and CCC. These are your next three letters, CCC and CCA. Let's check. CCC and CCC, CCA are both proline. That again is common to all the four sequences here. And then the next one is UGG, UGG, UGA. So UGG, UGA. So UGG is tryptophan and UGA is a stop codon. So we know UGA is a stop codon, so this sequence is eliminated. And UGG is common to all the other three sequences, so we still, all these sequences are still in the race. Now the next one is CAG. Okay, so CAG is glutamine. Okay, and CAC, the next one, CAC is histidine. So we have histidine here. So now we know that the sequence we are looking for is the last one, because this and this, they all get eliminated. So the answer for this question will be the last one, the last sequence, which is sequence number D. Please practice this. There you can have a lot of practice questions on the internet. Please make sure this concept is clear in your mind. Okay. Now the next question we will practice from the chart diagram, something like a box. Okay. So if you look at this chart, the tricky part here is, look, they have not given you an mRNA sequence, they have given you a DNA sequence. So you cannot work, so you have a T, you cannot work out with a T when you are looking for a genetic code. I said you have to look for an mRNA sequence. So if they give you a DNA sequence, you have to make it into an mRNA sequence. How will you make it into an mRNA sequence? Okay, I'll just make a box for you here, and let's start typing. So if it's 5' here, so I will have a 3' on my RNA sequence. Okay, and so first one is GCG. So for every G I have a C. I have to write the complementary sequence, then G and C. I hope you know this. Okay, I'm writing just a complement. For A I will write U, for C I will write G, and for A again one U, and for the third one TAC. So for T I will write A I will write U, and for C I will write G. And the next one ACT, A I will write U, for C I will write G, and for T I will write an A. So this is my mRNA sequence. First I will write my mRNA sequence, and then I will try to find out the amino acids. Look at the first one now, CGC. So C is the first letter, G is the second letter. This is the box I will look for. CGC is arginine. So my first amino acid is ARG. So what I will do here immediately is I will cut this alanine, because this is not my amino acid. Okay, now the next one is we have to choose from this. Now if we look at the second letter, we have UGU. U is the first letter, G we have to look in this box, and UGU is cysteine. The moment you know that, you know the answer. It has to be this one, because here there is no cysteine. So your answer would be answer number B. Okay, so I hope it is clear now how to read the genetic code and how to figure out what amino acid are they coding for. With this, I would like to stop this particular topic. In the next topic, we'll be starting off with the new lesson. Thank you so much for listening.